Hello. Thanks for watching our previous lessons. We are now starting new lesson series on biologicals medicinal products. This lesson 1 is an introduction to biopharmaceuticals. Here we will be learning on some general and brief topics under the introduction. Hope you will enjoy it. Learning objectives. In this lesson we will be covering definition, types of biotechnological products, properties of biopharmaceuticals, biopharmaceuticals structures. Further details will be presented in future lessons. However, as this is an introduction to the biologicals series, hence this lesson 1 will just provide a brief overview. Definition of biopharmaceuticals. A biopharmaceutical, also known as a biologic, medical product, or biologic, is any pharmaceutical drug product manufactured in, extracted from, or semi-synthesized from biological sources. Different from totally synthesized pharmaceuticals, they include vaccines, whole blood components, allergenics, somatic cells, gene therapies, tissues, recombinant therapeutic protein, and living medicines used in cell therapy. Biologics can be composed of sugars, proteins, nucleic acids, or complex combinations of these substances, or may be living cells or tissues. They, or their precursors or components, are isolated from living sources, human, animal, plant, fungal, or microbial. They can be used in both human and animal medicine. Types of biotechnological products is as follows: antibodies, recombinant protein peptides, blood and blood products, nucleic acids, vaccines, gene therapy, transgenics. Types of biotechnological products: antibodies, monoclonal antibodies, MAbs, antibodies fragments, scFv, Fabs, etc. Antibodies drug conjugates. A monoclonal antibody, MAb or MOAB, is an antibody made by cloning a unique white blood cell. All subsequent antibodies derived this way trace back to a unique parent cell. Recombinant proteins peptides. For example, recombinant insulin. Proteins are complex biomolecules that play various fundamental roles in living systems. They are made up of building blocks called amino acids that are attached to one another in chains. Blood and blood products. Blood products. Cellular components: red cell concentrates, platelet concentrates, and granulocyte concentrate. Plasma components: fresh frozen plasma, cryoprecipitate, cryopore plasma, stored plasma, plasma derivatives, albumin, immunoglobulin, coagulation factors, and blood products. Nucleic acids, DNA vaccines, RNA, aptamers, antisense oligonucleotides. Nucleic acids are the biopolymers, or large biomolecules, essential to all known forms of life. The term nucleic acid is the overall name for DNA and RNA. They are composed of nucleotides, which are the monomers made of 3 components: a 5-carbon sugar, a phosphate group, and a nitrogenous base. Types of biotechnological products: vaccines, gene therapy, transgenics. A vaccine is a biological preparation that provides active acquired immunity to a particular infectious disease. Gene therapy, also called human gene transfer, is a medical field which focuses on the utilization of the therapeutic delivery of nucleic acids into a patient's cells as a drug to treat disease. Transgenesis refers to the process of introducing an exogenous or modified gene, transgene, into a recipient organism of the same order and species from which the gene is derived. Properties of biopharmaceuticals. Biopharmaceuticals range in size and complexity from small monomeric proteins to large heteromultimeric protein complexes. Biopharmaceuticals structures can be divided into primary structure, secondary structure, tertiary structure, quaternary structure. Primary structure. Amino acid sequence of the polypeptide chain, held together by peptide, amide, bonds. Secondary structure. Highly regular local substructures, held in place by hydrogen bonding. Tertiary structure. Three-dimensional structure of a single protein molecule, driven by hydrophobic interaction, stabilized by specific tertiary interactions such as disulfide bonds, salt bridges, hydrogen bonds and the tight packing of side chains. Quaternary structure, larger assembly of several protein molecules or polypeptides chain. In our next video we will be learning about the further details on biopharmaceutical products. Thanks for watching and please subscribe for our latest videos.